Well, good morning, everybody. My name is David Wallace. I'm the director at the Newton Institute. I'm here just to say a few words of welcome to the Institute and to this workshop. Fantastic to see so many people who you guys have really hit the button as far as interest in this topic is concerned. This is a great example for me of the breadth of mathematical sciences that the Newton Institute cover. When I first came here almost a year ago, I had a fantasy that I would really be trying to interact meaningfully with every single program. And I failed miserably at that, largely because I've spent most of my time trying to raise money for the Institute, more or less successfully so far. But this is one where I really would be scratching at my backgrounds in theoretical physics and computing. But nevertheless, I will hope to have a chance to interact informally a bit with you. So welcome to the Institute. I hope that you find it a really conducive place for interacting. For those of you for whom this is the first time in the building, I hope it's a bit of an eye-opener. It's open plan. You're meant just to be able to come here to work and to interact. And I hope that the workshop works really well for you. So I think without anything more from me, I guess, over to Vincent, who's going to say a few words. And if you'll excuse me, it's my first day back from leave, and I'm going to have to scoot off to something. So I will leave you to it. All the best.
Thank you.
Yeah.
Okay, so thanks very much for all of you coming today. It's great to see so many people who are interested in this field. I just thought I'd pick through a few sort of short announcements here and say a few thanks. So this is sort of kicking off a 4-month program at this institute where we're going to have people from around the world studying and researching in phylogenetics. And this idea first came about actually in Sweden in 2004. So it's taken a little time to sort of bring this all together, but I think as we can see today, it's going to be well worth all of the effort, as it were. I'd like to thank the organisers of this of the actual program, Mike Steele and Daniel Hewson, for all of the help and all of the work they've done to make this program possible, and of course the Isaac Newton Institute for making this wonderful facility available to us. And we have various sponsors, but some of the people who are making major contributions include EPSRC, other than the Newton Institute, of course, include EPSRC, DSRC, and the Alan Wilson Centre. And I'm very pleased to also welcome some of the invited program participants who are here today and will be over here in the next 4 months. As for this workshop, of course, I would like to thank EMBO for their contribution towards making this workshop possible, and of course Isaac Newton Institute, and as well as Mike and Daniel, workshop organiser Brent Emerson. And of course, all of the invited speakers who will hopefully, well, almost surely give us some really interesting talks over the next few days. Now, if you can see over here, there's a conference dinner on Thursday. I believe most of you will have, if you have registered, you will know about that. That will be in your information packs. But if you would still like to go and you didn't register, then the deadline for that is by the end of the coffee break today. We've got, well, not we, there's a new video system in place here with 3 cameras, and I've been told they've just started to use that this week, okay? So they're getting used to this new system, and they've asked us, if possible, if we could make the changeovers between speakers rather carefully, so that when we put the microphone on the speaker, could we please do so and then announce the speaker rather than sort of doing it all at once. That will make it easier for them to keep nice videos of us. If you can, could you please put your talk on here, on this laptop in advance, if possible, okay? And you can just come up with the breaks and put your PowerPoints on, etc. And please, speakers and session chairs, I don't want to be too strict about this, but the lunches, etc., they're all kind of scheduled for certain times, so it would be very difficult for us to keep things going smoothly if we run over too long. So please try and keep to your talk slot and time. The Isaac Newton Institute has asked if you could please, while we're in the talks here, please turn off your mobiles and preferably don't use your laptops for sort of email and things. They prefer that you'll have your full concentration on the wonderful talks we'll be listening to. And there are a couple of questionnaires already, or at least one questionnaire that you should have already, and another one will be handed out, EMBO workshop questionnaire. I'd appreciate if you could please fill all of those in and return them. Hopefully it won't take too long, as it's sort of important for keeping the statistics. And if you have any questions at all, at the conference desk we have Chi and Tracy, who are very helpful, and I'm sure they'll be able to answer any of your questions. Okay, so please go see them. And if there's really any big problems, then of course come see me, and I'll try my best to help. So of course, hope that you'll enjoy the workshop. And now I'd like to announce our first speaker, who will be Tandy Warnow. But following what I've just said, I should not announce you properly until we've got your mic all fixed. Makes it a little trickier. Okay.
You want to introduce me?
Yeah, so our first speaker today will be Tandy Warnow, who's from the University of Texas at Austin, and she's going to tell us a little bit about computational and mathematical challenges involved in very large-scale phylogenetics. Thanks very much, Tandy.
Thank you. It's a real pleasure to be here. Can everyone hear me? Okay. So everyone here knows about phylogeny, but it's interesting to think about it not just in terms of small data sets, but large data sets, and also what the phylogenetic analyses can do for other kinds of research. There's a lot of biomedical applications. I personally don't work in the applications, but the computational challenges instead. So I'm going to be focusing on the computational challenges. So the steps in the phylogenetic analysis. It's interesting to think about how much time goes into these and what's involved. So first of all, the biologist goes out into the field and collects data, and that can take a very, very long time. And it's good for mathematicians to appreciate that the amount of time that they spend doing the analysis is actually much smaller than the amount of time they spend getting the data. It's an interesting challenge. Then they align the sequences. That step itself can take a long time. Reconstruction of the phylogeny from the aligned sequences is what we normally look at. This is normally the computational problem that we're concerned with. Once you have an alignment, how do you get the phylogeny? When you do that, if you're solving maximum parsimony or some other optimization problem, you can get a large number of trees. From that set of trees, you're trying to estimate what's reliable about the evolutionary history. That can be done in various techniques, for example, through consensus methods, but also through statistical techniques. And after you've done that and you have an estimate of what's reliable about the history, then you do post-tree analyses, which are trying to answer biological questions. Now, the reconstruction of the tree of life itself has many complexities. First of all, just computational. Just about every problem that is involved is NP-hard, which means it's hard to solve exactly, and you end up resorting to heuristics. And if you really wanted to do the tree of life, you'd have 8 million sequences to look at, approximately. And that's a large number. Trying to solve a hard problem on a big data set can be really enormously difficult. But evolution is also very complicated. It's reticulate, which means you have horizontal gene transfer or hybridization. There are events that happen in evolution that are not generally part of the models of evolution that we look at. These events include insertions and deletions of strings. They include rearrangement events that change the order of the genes within the genomes and the number of copies of each gene. And there's also differences between the trees you get for individual markers and the tree that you want to estimate on the species. And so your estimates of the history of what you're looking at may not match the species evolution. So trying to get a species tree is harder than just getting the different gene trees. All of this is part of what makes this area very interesting from a mathematics or computer science standpoint. Now, the Cypress project is a project that's funded by the National Science Foundation in the US. And I had this project, and it has a number of different components. And it's all geared at enabling large-scale phylogenetic analysis. So we develop algorithms and software focusing on large data sets. We have fundamental research in mathematics and probability related to Markov models of evolution and optimization problems. We work on the problem of storing and relating different trees to data sets and to each other. Tree-based is something that we are helping to develop new versions of. Simulations, we have the first million taxon, million site simulation under complex evolution. We do a lot of museum outreach and K through 12 outreach. And we have a portal available at the Cypress website if you want to do analyses using Cypress software. So you can just go to our webpage, www.philo.org, and upload your data sets and get back analyses. Now, this talk is going to focus on mathematical and computational problems, and so it has 4 parts. The first part is an overview of some interesting mathematics questions and results primarily related to Markov models of evolution. The second part is looking at how do we approach trying to solve NP-hard optimization problems. Now, when I say solve, I really don't mean solve, because these are problems that are essentially, forget trying to solve them exactly. So we develop heuristics, and how do we evaluate heuristics, and how do we try to make better performing heuristics. The third part is new work that is not yet published on how to simultaneously estimate a phylogeny and an alignment from unaligned sequences. And I'll show you some preliminary work there. And then I'll conclude with related research and some open problems. So the first part, mathematical issues under stochastic models. So this is the usual picture of how a DNA sequence evolves without insertions and deletions or rearrangements, just the usual models where it's just site substitutions. You have a sequence at the root, and this is a speciation event, so it branches into 2. And there are, in red, I've highlighted the positions that have changed. So you're seeing site substitutions. More speciation, more site substitutions, more speciation, and more site substitutions. So this process starts at the root of the tree with a sequence, and that sequence evolves down the tree, and the entire process is modeling just site substitutions, okay? And if you treat that in the usual way, then you're treating all the sites as operating independently of each other. And so we can talk about trying to reconstruct the tree from the sequences that we see at the leaves of the tree, and that's the usual Markov model approach to trying to estimate phylogeny. So you get to see what's at the leaves, but you don't get to see what the tree looks like, and you don't get to see the ancestral sequences. Now, Markov models of single site evolution, they range from the simplest DNA model, which is the Jukes-Kanner, to the most complex single site evolution model, which is the general Markov model. The simplest one is what you would expect it to be. The simplest one, all the sites are independently evolving identically under the same process. If you change with equal probability to the other nucleotides. And the nucleotide at the root is random. So that's the simplest of the 4-state models. And you can make it more complicated by having the different nucleotide substitutions having different probabilities. But again, that's a single site evolution. And the question is, what happens when you try to incorporate the differences between sites? So there's something called the rates across sites assumption, which is very standard. And what it's saying is that if you draw the tree for one site with branch lengths proportional to the expected number of changes, then the 2 trees for different sites will just look like scaled up or down versions of each other. So this is supposed to look like 2 different sites, and they're supposed to be scaled, so all the proportions are the same. And so what that would mean is that if one site is twice as fast as another site on one edge, it's twice as fast everywhere. So that's the rates across sites assumption. And then you would also assume that you're drawing those rates from a distribution. And if you make the distribution of where you draw those rates nice, then you have nice properties about your model, and you can try to reconstruct the tree from observed data. So the usual assumption is that the rates are drawn from a gamma distribution. But that's the rates across sites assumption. Now, you can ask whether or not it's realistic. After all, different environments might make different sites speed up or slow down differently, not all exactly the same. And that could lead you to what's called the no common mechanism model, which is to say that you have an underlying tree that's fixed, but now one site and another site, they don't have this kind of scaled up or down relationship between them. So one site could be twice as fast as another site on one edge, but only 1.5 times as fast on a different edge, or even slower. So that's the no common mechanism model. Now, identifiability is a very fundamental concept about phylogeny models and about models in general. And in the context of phylogeny, what it's saying is that a model is identifiable if you can actually determine it uniquely from the probability distribution that it defines. So you can think about one of these Markov models as defining a probability distribution of nucleotides at the leaves. So with a certain probability, you'll have all A's. With another probability, you'll have all T's. With some probability, you'll have half of them A's and the other half T's, particular half A and the other half T's. From those probabilities, you could determine the tree. That would mean that the model is identifiable. Now, a reconstruction method is statistically consistent under the model if you can guarantee that that method will reconstruct the true tree with probability going to 1 as the sequence length increases. And so what that's saying is that as the method is able to get better and better estimates of those probabilities of each of the patterns, it would actually be able to reconstruct the tree with probability going to 1. So the different property, one is about the model, the other one is about the method. And if a model is not identifiable, then you can't have a statistically consistent method. So identifiability results, you will hear some new results in this conference. But the standard Markov models that people work with in phylogeny are identifiable. That doesn't mean that all models are identifiable, just the ones that people like to work with. If you draw rates from a nice distribution, then you can also have identifiability. But if you draw it from a distribution that's not so nice, then you don't have identifiability. Mixed models are often not identifiable. So there are 2 talks later in the conference that you should hear that will be addressing these issues, Allman's talk and Matson's talk. Very interesting new work. So even in the fundamental questions about identifiability, there's some new results coming up and open questions. Now, phylogeny estimation is almost always done under identifiable models, and all the software that people use fundamentally is for identifiable models. Now, what about phylogeny reconstruction methods? Because after all, proving that something's identifiable doesn't tell us that we can get a good estimate of the history from the data. So what are the methods that people use to reconstruct trees from data? So this is a cartoon of you have sequences at, just given to you, and you want to reconstruct the tree. And what you're reconstructing here is an unrooted tree, even though it's a rooted tree that generates the data. And that's generally the case that in reconstruction methods, because even though the model's trees are rooted, you can't locate the root. So the best you can get is the correct underlying unrooted tree. And if you get that part correct, then you're determined to have a correct reconstruction. There's basically 3 classes of methods. There are hill climbing heuristics for NP-hard optimization problems. Sometimes you can solve those exactly, but people rarely can afford to use exact solutions because they're limited to very small data sets. And it's easy to get stuck in local optima where all of your moves away from where you are make the score worse, and so you just stay there. So optimization problems that are NP-hard are very hard to solve because of these local optima. Polynomial time distance-based methods is another class. These are methods that will complete in polynomial time and give you a definite answer. And then there are Bayesian methods typically based upon MCMC. So roughly 3 classes of methods, and only the second class is polynomial time. The first and the third are computationally intensive. Performance criteria. If you're a computer scientist, you would think about running time in space. If you're a statistician, you would think about statistical performance issues such as statistical consistency, which we've talked about, but also how much data the method needs to reconstruct the tree with high probability of success. So that's what we call the sequence length requirement. Now, topological accuracy can be measured in simulation. So you can look at results without proving theorems. You can look at performance in simulation and compare methods to each other. And that's the standard technique in the literature, is to do simulations. You can also say, well, I'm interested in solving a certain optimization problem. How well do the methods work with respect to that optimization problem? And that you would study either in simulation or on real data. So different ways of approaching understanding methods. Real data you can study, you can use in some ways. Simulated data you can use in others. And you can prove theorems. Okay? How do you quantify accuracy? There is, as I said, you're reconstructing the unrooted tree from data generated by a rooted tree. So this is a cartoon of what a simulation study might look like. You start with the true tree, and it's rooted, and you generate data, S1 through S5, and then you reconstruct the tree. Now, this tree that you've reconstructed is not the unrooted version of the true tree, and you can tell that by just looking at the edges. So, I don't know how to use this. There. This edge splits S3 and S5 from S1, S2, and S4. There's no edge here that corresponds to that. So I've marked it FP, which stands for false positive. On the other hand, this edge here splits S1 and S2 from S3, S4, and S5. And so does this edge. So that edge is not marked because it corresponds to an edge in the true tree. Okay. Similarly, this edge here splits S4 and S5 from S1, 2, 3, and there's no edge in the inferred tree that matches that. So you can mark the edges as being false positives. They show up in your estimated tree, but they shouldn't. False negatives, they're in your true tree, and they don't show up in your estimation. And the other edges are correct edges. Okay? So if you have both trees binary, then the false negative and the false positive rates are the same. And otherwise, you can actually talk about two different kinds of error rates. So this is like sensitivity and specificity. Okay? So in this case, it's a 50% error rate because one edge is right and one edge is wrong. Both of them are 50%. Okay? So that's how you measure error. This is a cartoon of what statistical consistency implies and what the convergence rate implies. So if this is a simulation study, I might look at one model tree, simulate sequences on that model tree of a certain length, and give that data to two different methods. So let's just talk about one method. I give that data to one method, and I get a reconstruction, and I score it for how accurate it is, and I record the error rate. And then I take longer sequences, and I do the same thing. And then I take longer sequences, and I do the same thing. And as the sequence length increases, you will expect to see the error rate eventually going to 0. Now, it may not be monotonically going to 0. In this case, they're both monotonically. But these are two different curves for two different methods. Both of them eventually go to 0. And that's showing statistical consistency on that model tree, because the error eventually goes to 0. But the question is, at what point does it go to 0? How much data does it need to go to 0? This method, this curve, is going to 0 faster than this one is. This one is going to 0, but it takes longer. So if this one goes to 0 from a polynomial number of sites, then we would call that absolute fast converging. And if it goes to 0 from an exponential number of sites, we call it exponentially converging. So you're talking about how much data it needs to go to 0 with high probability, and describing that as a function of the number of taxa. It's also going to be a function of the parameters of the tree, but if you hold those other parameters fixed, you can talk about it as a function of the number of taxa. So what are the theoretical results about statistical consistency? Neighbor joining is polynomial time. Neighbor joining is the most popular distance-based method, and it's polynomial time, and it is statistically consistent under the typical models that people work with. Maximum parsimony is an NP-hard optimization problem, and even exact solutions are not statistically consistent under the standard models. Now, maximum likelihood is NP-hard also, and exact solutions are statistically consistent under these typical models. Now, I'm trying to emphasize exact solutions because it's an NP-hard problem. You can't solve it exactly. And so you're stuck with the issue of the fact that you're working with heuristics and the guarantees you have about performance for maximum likelihood do not apply to your heuristic analyses. What about sequence length requirements? Here, the results are much more recent than the results about statistical consistency. Neighbor joining, it's statistically consistent, so we can talk about its sequence length requirement. And unfortunately, it, as well as a lot of other distance-based methods, requires sequence lengths. Well, I shouldn't say requires, but the sequence lengths that we know suffice for accuracy grow exponentially in the diameter of the tree. Now, that's a problem because the diameter can be as bad as order n, and it's typically square root of n. So this is not a very, very fast convergence in the sense of getting the true tree from, let's say, polynomial length sequences. Maximum likelihood, the best bounds we have right now are worse than the bounds for neighbor joining. That's kind of surprising because if you look at simulations, maximum likelihood will generally do better than neighbor joining. But this is the current theory. It's limited to this kind of a result. Okay? Now, what about in practice? This is a simulation study that we performed that looked at the error rates we got by fixing the sequence length but taking bigger and bigger model trees. And so the model trees are random trees with branch lengths all drawn from a particular distribution. And you just get bigger and bigger trees, and as you get bigger and bigger trees, you also have bigger and bigger diameters because of the way we're making these random trees. We evolved Chimera 2-parameter model sequences down the tree. First, the root has 1,000 sites, so everything has 1,000 sites. And we looked at the error. And what you see here is that as the number of taxa increases, the error rate increases. Now, this is 20% of the edges are missing. 40% of the true tree are missing. So this is really bad, right? This is not as bad as random trees, but this is, you're missing 40% of the edges of the true tree. Now, the most you can have is 100% missing, so this is going up pretty bad. Okay, now you might say, well, the way you're generating your data, there's so much homoplazing that it's a problem of the data. You can't get a better tree. You only have 1,000 sites. There's so much evolution going on, you can't get a better estimate. The problem is the data is not the method. Now, I'm going to show you that that's not true, but let's just leave that as a conjecture. So we developed a method called DCM1, and Catherine St. John is in the audience. She's one of the authors on this. This is a two-phase method. The first phase is DCM, and the second phase is SQS. So DCM what it does is it takes your data set and it breaks it up into a collection of smaller overlapping subsets. It passes each of those subsets to your base method, which will then reconstruct a tree on each of those subsets. It then combines those subtrees into a tree on the full data set. And it does that process n squared times. So it's n squared different decompositions and therefore n squared different trees. So that's what comes out of the DCM phase, is n squared trees. In the SQS phase, we pick a tree that optimizes the criterion that SQS stands for. So the first phase generates n squared trees. The second phase, you pick the tree that optimizes that criterion. And the theorem says that if you put in an exponentially converging method as your input, in other words, your input is a method, like neighbor joining, and what comes out of this is another method. This is a meta method that takes a base method and turns it into a method that is applying the base method to subproblems. So if you start with an exponentially converging method for your base method, what you get is something that's absolute fast converging. And remember, absolute fast converging means that its polynomial sequence lengths will suffice for accuracy. So this result produces this empirical performance. Same experiment that I showed you earlier. But now, instead of running neighbor joining just on the full data set, we run neighbor joining just on subproblems. And the amazing thing is that the error rate has not yet started to increase. It will eventually increase. But the error rate is, just staying down at 8% approximately, which is quite surprising. So it's not the data. It's the method. And that's one of the lessons, that you can improve things by changing your method. Now, the problem is that in practice, these methods don't perform as well as for example, maximum likelihood. And there's still a community that wants to use maximum parsimony. So what you have is that what people really want to solve is either NP-hard maximum likelihood or NP-hard maximum parsimony, or else they want to run Bayesian methods. And all of these methods are computationally intensive. And so despite the nice theory that we have, we still have, as a challenge, a fundamental challenge is how do we address the need to solve these hard optimization problems? So the second part of the talk is how do we improve heuristics for NP-hard optimization problems? Standard approaches for solving them combine hill climbing, but the hill climbing can get stuck in local optima, with randomization to get out of the local optima. So that's the standard heuristic design. There's actually very little that's really understood theoretically about the performance of these methods. So we can study these things on real data, we can study them in simulation, but we don't really know how to prove theorems about the approximation ratios that these approaches can give us. But they perform very well, and they perform better in practice than approximation algorithms with known guarantees. However, even in practice on big enough data sets, even the best methods can take quite a long time to recover sufficiently accurate solutions. So now we're talking about how do we measure how good an NP solution might be. Well, if suppose this is an example of what we did. We took a number of different data sets and we ran many different methods on those data sets, and we let them run for as long as we wanted to, which in some cases could be months. And we recorded the best scores that could be found on those data sets. And so we're calling that optimal, but optimal is really in quotes. It's the best score to date on the data set found by any method. So this is an example of running a TNT heuristic. TNT is actually the fastest maximum parsimony software that you can get. Running it on a data set with almost 14,000 sequences and recording the best score, and that's the target. Now, it turns out empirically that if you have scores that are more than 1/100% bigger, then the topologies of the trees that you're getting can be quite different. So you really need an accuracy that's within 1/100% of parsimony score to get approximately the right tree back. In other words, topologically no more than about 7% off. Okay? So how well does that heuristic work? Well, this is recording its drop in error over time. So after like 2 hours, we recorded the parsimony score we found at 2 hours, and the parsimony score we found at 3 hours, and parsimony score we found at 4 hours. And that score is dropping, right? And it's approaching the best score that can be found on the data set, but it's approaching it very slowly. As you can see, here it looks like there's nothing left to find. Then it drops again, then it drops again. But this is at 24 hours, and we need to get down here. It turns out that this particular heuristic on this particular data set was going to take months. So it looks like it's converged, but it really hasn't. So the observations that we had is that the best parsimony heuristics cannot get acceptably good solutions within 24 hours on some large data sets. And data sets of these sizes using existing techniques seem to need months or perhaps years of additional analyses to solve them within reasonable levels of accuracy, not exactly. And apparent convergence can be misleading. So we designed a technique for improving these heuristics, and it's a similar technique to what we used for DCM1. You remember DCM1 we used with neighbor joining to improve its sequence length requirement. We designed a similar thing for running with maximum parsimony, and then we subsequently also used it with maximum likelihood. And so basically what it does is it has only the first phase. The first phase is where you do a decomposition, you apply your base method to the subsets, then you merge the trees into a tree on the full data set. But we combined that with a different technique for the decomposition, and we combined it with iteration so that we could actually use it repeatedly as a way of getting out of local optima. So the decomposition technique was what we call DCM3. All of the DCMs, DCM stands for discovering method, all of the DCMs are graph theoretic decompositions which exploit theorems about triangulated graphs in order to get both their running time and their guarantees. This is an example. So suppose we have a current tree that we're looking at. So we're doing a maximum parsimony search. We get stuck in a local optima. That tree becomes the guide tree. On the basis of that tree, we construct a graph which is called the short the short subtree graph. Now that short subtree graph in construction only takes order n time, but it's triangulated. And triangulated graphs have very nice properties, including that you can find all of the maximal cliques in polynomial time. So from that short subtree graph, we compute a clique separator and use that clique separator to divide the graph into approximately 4 or 5 subproblems. That collection of subproblems are then fed to the base method. And if we apply that decomposition again, then we're applying it recursively. So this decomposition produces a triangulated graph. The clique separator decomposition gives us the decomposition into subproblems, and we can apply it recursively. We can then construct trees on the subtrees, trees on the subgraphs, combine the subtrees into a tree on the full data set, and then resume the hill climbing. And then when we get to another local optimum, we can repeat this process. So this is an example of how we use it iteratively. We have some initial tree that we get, and we do hill climbing using our base method, and we get to a tree here. And then we stop and we do the decomposition and apply the base method on the subproblems and combine the subtrees to the full data set and get T, and then we hill climb again. We get T prime. Now, the base method may not be hill climbing. It may be something more complicated, but we're alternating between applying the base method on the full data set and applying the base method only on subsets. And that alternation is how we get out of local optima. What's nice is that this combination significantly improved upon the parsimony heuristics that we looked at. And this is an example on that same data set that I showed you before. The score is dropping using TNT, and the score is dropping using the REC IDCM3 version of TNT. So we're getting to much better scores much faster at every point in time that we were scoring. And what we saw, we looked at a lot of different data sets, and what we saw is that the improvement you got would depend upon the difficulty of the data set. If the data set is easy to solve, you don't get any improvement. But as the data set gets harder and harder to solve, you get more and more improvement. So the general observations is that we improved upon the best performing heuristics for maximum parsimony, and that the improvement would increase with the difficulty of the data set. Subsequently, we have implemented this also for maximum likelihood, and it's available through the Cypress portal. So you can get very fast analyses of maximum likelihood analyses of large data sets through Cypress and through the portal. The software is also available for download. Now, all of this was about what do you do when you have a data set that's already aligned. What I'm going to talk about now is how do you deal with the problem when the data are not aligned. So this is new work. Remember the steps in a phylogenetic analysis. First you go out and you get the data. And then you align the data, and then from the aligned data, you reconstruct the tree, and then you do something to estimate what's reliable about the evolutionary history, and then you do your post-tree analysis. So what I'm really interested in talking about here are these two steps, aligning the sequences and then reconstructing the tree. So starting with unaligned data to getting the tree. This is the standard approach. It has 2 phases. First align, then get the tree. Okay? It's not the only way to do it, but this is the standard way. So the basic observation about what we call this as a standard 2-phase method, the standard thing for a systematist generally is to use the software called Clustal, one of the Clustal variants, Clustal W, whatever, and then take that alignment and fix it if you can, and then take the final alignment and run it through parsimony or Mr. Bayes or a likelihood approach. Okay? So Clustal is a standard alignment routine that most systematists would work with. But the protein people, which is to say people who study proteins, have been working on alignment methods on their own and come up with a whole new collection of alignment methods that haven't really been tested in their phylogenetic context. But they perform better for protein alignment. And we've tested them, and they actually perform better for phylogeny purposes as well. And so you can actually do much better by moving from Clustal to one of the newer alignment methods. In particular, ProbCons and MAFT are the 2 best performing of the alignment routines. Now, alignment accuracy can be measured in various ways, but the standard ways that the protein alignment community has worked with is called the SP score. And that SP score does seem to correlate with phylogenetic accuracy, which is to say, if the alignment has a better SP score, in general, you're going to get a better tree. But it doesn't seem to produce as big an impact as you might expect. So big improvements in SP score might not produce big improvements in the tree. And this is unpublished data. But what about simultaneous estimation? I mean, probably many of you have heard about other techniques for doing estimation of trees from unaligned data, of which there's statistical methods and then the extensions of parsimony. Now, the statistical methods, they have a lot of appeal because statistical phylogeny reconstruction definitely outperforms parsimony. I mean, you can get much better results when you look at large data sets. It's not true on every model condition, but in a lot of model conditions, you get much better results from maximum likelihood. Why not try to do maximum likelihood or a Bayesian approach for the simultaneous estimation of trees and alignments? The problem is that although some methods have been developed, they don't scale. The biggest data set that any of these methods have really been able to analyze is maybe 20 or 25 sequences, and even that has taken, a week or something. It's really not clear that those approaches can scale in their current implementations. The extension of parsimony to this case is where instead of just counting site substitutions, you would also use the gaps in your calculation of the tree length. And so the cost of a gap would be some function, typically an affine function of the gap length. And how do you solve the minimum tree length problem there? Well, that's an NP-hard problem also. The software that's been developed to solve that, POI, can be run on data sets that are large. But two things. One is it takes a very, very, very long time. And secondly, in practice, it doesn't give you trees that are as accurate as the best two-phase techniques. So in the current situation with simultaneous methods, some of the methods just don't scale, and the others don't perform well. So that's the current problem. Now, we have a new method. This is unpublished, my group at Texas. And it's basically a technique which is trying to combine insights from both communities. So instead of being a Bayesian technique for the simultaneous estimation of alignments in trees, or instead of trying to do maximum likelihood under models that include gaps, we're doing something different. We're proposing alignments on the basis of trying to solve the minimum tree length problem. We're solving the minimum tree length problem using a heuristic, and we're using carefully selected affine gap penalty for the data. Then we propose a collection of alignments, and each alignment is then used to solve maximum likelihood under GTR. So it's not under a gap-based model of evolution, but just under the GTR model. So we run RAXML, which is software for maximum likelihood, on each of those alignments that we propose. So this technique is what I'm going to show you. The simulation study that we performed, we've actually looked at 25 taxon trees, 100 taxon trees, and some 500 taxon trees. And so we're really trying to go for large data sets. And what I'm going to show you is our results on 100 taxon trees. So with 100 taxon trees, we generated the model trees using the rates software and then deviated the branch lengths so that we were no longer within the molecular clock. And we simulated evolution using the rows software, which produces gaps, so insertions and deletions, under two different gap length distributions and two different probabilities of gaps and two different probabilities of substitution. So we have in total 8 different model conditions. Okay? And these model conditions range from not very gappy to very gappy, short gaps to long gaps, and high rates of evolution and moderate rates of evolution. And then for each of the data sets that we obtained, we recorded the true alignment, which we know because we're running it in simulation, and we computed MAFT and ProbCons and Clustal alignments. And then we took all of those alignments, the 4 alignments, and gave them to RAXML to get trees. And we're scoring the alignments for accuracy in various ways, and we're scoring the trees for accuracy. Oh, no. This is a problem. it's PowerPoint. Can you get my Mac out of my office? M4. My apologies. It shows up on my Mac, but it does not show up here. Daniel is going to come get my Mac. Someone have a Mac down here? Can I have your Mac? Does it need another connection? Is it on? I left my memory stick upstairs, I think. How do I connect this? Can we connect this to the internet?
I'll go and get your presentation on the
Well, well, this is a memory stick. It's not, okay. This is crazy. I should have checked. Ah, is that's not mine.
That's not yours.
No.
How many mix does the Texan need?
It's the Warnows up there. Right there. Just so I hope they edit out the video.
This won't work now. Okay.
I don't need it to work. Yeah. Warnow. Yeah. Yeah. This is crazy. So I owe you coffee.
All right, because I need.
I'd be very happy. Very.
Should we actually we go to the
So why is it not moving?
it will
You know what? You need to like bring this up though.
go straight.
Let me just do it, okay? Thank you. Thank you, thank you. Okay. Make sure your other stickers make sure that their talks show up.
Yeah, that's good.
Not quite as safety off for you.
It's okay, it's okay. Just
Let's just hit the. Oh no, you've gone back to the start.
oh no.
There you go, that's good.
Right, it wasn't good.
That was good.
Go to the. Go to the.
No, no,
What's good?
Go down.
Slide on
no, that was not the problem. The problem is the screen resolution was bad.
Thing again, it was good.
Screen resolution was
It
bad.
changed. Let's do the display thing again.
It's fine.
Okay. Okay, so
That's okay, go full screen. Should be okay.
okay. It's a good thing I was talking quickly. Now I've saved some time and I used it up. Okay, so simulation study. 100 taxon model trees, 8 different model conditions, long gaps and short gaps. And when I show you the results, the first 4 models will be the long gap models, and the second 4 model conditions will be the short gap models. And we computed Raxamel trees on each of the alignments that we obtained. And the first thing I'm going to show you is alignment accuracy, so tree accuracy. So the model conditions are on the bottom, 1 through 8. You can see that 3 and 4 and 7 and 8 are the hard model conditions because these are the cases where the error rates are the highest. Now, in each case, from left to right, it goes from Raxamel on the true alignment, then Sate, our new method, then Raxamel on ProbCons, Raxamel on MAFT, and finally Raxamel on Clustal. So in this model condition, almost all the trees have 5% error or less. And Raxamel in the true alignment is basically the best you can do in this approach, right? Because you're getting maximum likelihood on the true alignment. So this is an easy model condition. They're all doing well. This is another easy model condition. They're all doing well, although you're starting to get more error from Clustal. This is a model condition where you can see Clustal having a really hard time. But Raxamel in the true alignment and Sate are almost the same, and then these methods are a little bit worse, okay? This is similar to here. Here, similar to before, same as before. 7 and 8 are the hardest conditions. So 7 and 8 are short gaps instead of long gaps, and very gappy matrices, something like 70 or 80% of the matrix is gapped. And the maximum normalized Hamming distance is about 55%. So it's not that bad for how much evolution is happening in terms of site substitutions, but there's a lot of gaps being introduced. And what we're seeing here is that Raxamel in the true alignment is getting about 5% error. We're a little bit higher. We're about 7 or 8% error, but we're close, whereas all the other methods are doing much worse on 7 and 8. So we're getting a big improvement over the best current 2-phase methods, and we're getting close to the benchmark, which is Raxamel on the true alignment. So that's the topological accuracy improvement that we're obtaining using these techniques. What about alignment accuracy? Well, one thing you might look at is, you know, the true alignment length, which is the number of columns in the true alignment. How well does the method reconstruct that? In other words, produces an alignment with the right number of columns. This is all normalized by the correct number of columns. And we're looking at the true alignment, Sate, ProbCons, MAFT, and Clustal. So the best you can get is 1, right? So the true alignment always gets 1. In some cases, we're a little bit more than 1. We have a slightly longer alignment than you would expect. And in some cases, we're below. But we're much closer to the true alignment length than ProbCons or MAFT or Clustal. And what you can see is that Clustal in particular has a tendency to really compress the data. Okay, now part of the problem with all of these methods is that they've been optimized to be good for what is called the SP score. And the SP score, which I'll show next, is a measure that only says what you care about is getting the correct paired nucleotides. So if there's a nucleotide homology pair in the true alignment, you want to get that in your estimated alignment. So you're not being penalized for incorrect pairs. You're only being penalized for missing pairs. So you're going to end up compressing stuff because you don't lose anything by compressing. Here's what you get when you look at those metrics. FN corresponds to what people call SP, but we're putting it as an error metric rather than as a good thing. So it's the complement of the SP error. And again, for the Sate alignment, the ProbCons alignment, MAFT alignment, and Clustal alignment, so we look at 2 types of alignment errors in this case. The SP error, which is the missing homologous pairs from the true alignment, and the false positive, which are the pairs that you put as homologies that are not, okay? And you can see that almost 100% accurate alignments here and here, except for Clustal. Here, Clustal is much worse than the others. Again, Clustal is much worse. And in general, Clustal tends to be the worst. But what's interesting is that we are not the best. You know, Sate, although it got the best trees, isn't giving the best alignment scores with respect to this metric. So it is the best in some of the cases, but it's not the best here or here. So it's interesting. I mean, this SP metric, whether you do it as false negative or false positive, it's not necessarily predictive of the phylogenetic accuracy that you're looking for. And the basic problem is you're not scoring the pairs, the correct pairs of nucleotides with gaps. So you're not keeping all of the pair information around. So the observations are Satay is more accurate at estimating the correct number of gaps and the locations of those gaps and doing the correct alignment when you have very gappy story going on. It's comparable to the other methods when the alignments are not that hard. You get a comparably good alignment as ProbCons and MAFT under the easier conditions. But when the model has a lot of gaps in it, you're going to get a much better estimate of the alignment and much better estimate of the tree by running Satay. That's where it really provides an advantage. SP as a metric is not particularly predictive of the alignment accuracy with respect to phylogeny. So from a phylogenetic perspective, we really have to be concerned about claims of improvement of alignment methods over other alignments just purely on the basis of this SP metric. And we still need alignment methods that can handle rearrangements and duplications. All of the alignment routines that people use really don't handle those more complicated events. And certainly when you want to work on large data sets, you just don't have such techniques available to you. So then part 4 is the open questions. And luckily it's short, so I'm almost done. Tree shape is something that people occasionally study in terms of its impact on phylogenetic accuracy. But we work a lot on models of sequence evolution, and we don't work that much on models of cladogenesis. And so what are the good models? I mean, is the birth-death model really the right thing to be working with? I mean, years ago, David Algis gave a very interesting talk about the beta splitting model, which is an alternate approach. And there are other approaches that people have also posited, but we really don't have a good handle on what are some good cladogenesis models and how they impact phylogenetic reconstruction. So that's another research area that I haven't even touched upon. As I said earlier, maximum likelihoods sequence length requirement, the current bound is worse than the bound for neighbor joining. And yet no one would really believe that, although maybe I shouldn't say no one. I mean, there is a chance that there are people who are right and that they believe otherwise. So it's interesting. It's not trivial. And it's not obvious what the answer should be. What is the correct sequence length requirement for maximum likelihood? A third question is, why is maximum parsimony not so bad? I mean, it's not statistically consistent. You don't, and if you do simulations, you don't, in fact, see that it's reconstructing the true tree. And yet, its error rate tends to be relatively low. So why? I mean, can we prove some theorem about this? Can we prove something about, what percentage of the edges we should be able to reconstruct, or which kinds of edges we should be able to reconstruct and under what model conditions? I really would like to understand this better. Another question which various people will be talking about during this week is the reconstruction and detection of reticulate evolution. So how do we do that? How do we detect horizontal gene transfer? How do we locate it correctly? So how do we construct really accurate evolutionary scenarios that involve reticulation? It's very interesting and very hard. Teasing apart two trees, that's a question where you have two trees and you have data. And you're told one of these two trees is the true tree, but now you have to figure out which one it is. So how hard is that problem? It should be clear why it's interesting. Because often in an analysis, you're going to get a multiple collection of trees, and you have to figure out which one of those trees you should have confidence in. If you could tease apart two trees, you would be able to select which one you want to keep. And we had that kind of approach with our DCM1, right? The second phase picks one tree out of a collection. Now, how much data how do you design methods that can do this well? And how much data do they need to answer these questions? This is something that you can ask Mike Steele about. He has some results in this area, and very interesting results that need to be extended. And finally, what complex models are identifiable? We know about GTR being identifiable. What else do we know? The general Markov model is identifiable, but what else do we know? You'll hear about GTR plus gamma later on, but what about models that involve insertions and deletions and rearrangements? What about other distributions of rates across sites? general comments that I would like to make are almost all the theory that we talk about is for identifiable models. But these models are clearly, the ones that we work with are clearly too simple. And their simplicity, what I mean by they're too simple, is that we know they don't match biology. The reasons that people like to work with these models is two things. One is they are identifiable, so you have some nice guarantees about what you can reconstruct. Secondly, you can develop inference methods that can be efficient. But that doesn't necessarily make them the right thing to do. And even if they are the right thing to do for inference, if we're going to understand their performance on real data, we actually need to simulate under much more complex models. Or we need to prove theorems about what happens when the data come from a complex model, but you estimate under a simple model. So I think it really behooves us and the biologists that we know to develop models that are statistically appropriate for the data, and then study the inference problem under these models, even if the inference methods are under simple models. Relative performance between methods, reconstruction methods, changes as you change the model, and they change as you go from smaller data sets to larger data sets. So all of the confidence that we have that we understand the performance of methods because we looked at 20 taxon trees doesn't hold. It's one of the things we discover when we do these simulations on bigger data sets. The relative performance changes. Neighbor joining can be better than parsimony on small data sets, but when you look at larger data sets and you don't have extremely long sequences, the relative performance can change. So you really have to look at both complexity and the number of taxa. And we really don't know how well methods perform under realistic conditions, either with respect to the model that's generating the data, or do we know how long we have to let the methods run to get a reasonable analysis. Both of those things we don't know. Acknowledgements to my funding and my collaborators, of whom a few of my collaborators are in the audience. I want to thank them for the times we've had. And that's it. Thank you.
Any questions? Any?
So 2 questions. First, you mentioned that for MP, for maximum parsimony, some algorithms with provable approximation bounds are better starting point. Is there anything? I thought you could say it. Maybe
I didn't say that, no.
Okay, sorry. Okay, but anyway, they exist. Do you know anything like that for maximum likelihood? Is there anything for log likelihood or something?
No, it's trying to come up with bounds on log likelihood would be a really good thing to do, and I don't have any information on that.
Okay, and the second is a comment. So all these NP hardness things, so most of them, the data that you're producing to prove hardness are really very non-tree-like at all. So is there anything we can say, okay, in this region, okay, it's hard, but in the realistic region, maybe things are better. Is anything like that?
You know, that's a really interesting question, and one that I don't have any answers. I don't think anyone knows yet, but one of the things I would say is that when you try to solve maximum likelihood or maximum parsimony on simulated data, it's a lot easier than running it on real data, which would suggest that perhaps the models that we're using to simulate data don't really match reality all that well. But it also might suggest that, in fact, the problems are not as hard under the models that we're generating the data. I mean, both things, right? But I don't think there's any theorems yet along those lines. I mean, that is the kind of thing that AI people like to look at, right? They like to look at what kinds of classes of data sets within NP-hard problems remain hard and which ones become easy. But I don't know any such results in parsimony or likelihood.
Tandy, you mentioned at the beginning that the tree of life would be on 8 million species. Now, if I look at NCBI, there's roughly 190,000 different species represented by sequence at the moment, on the one hand. If I look at nematodes, the number of different species of nematodes is estimated to be between 1 and 100 million. So where does the number 8 million come from?
8 million is the number not of species, but of the subset of a species that people think would be sufficient to estimate the tree of life. That's a number that's sort of just the atoll community in the US sort of says that's sort of the standard number that they're approximately talking about.
Any other questions?
You showed some results for performance of neighbour joining with large numbers of taxa. When neighbour joining is doing really badly with big trees, which edges is it getting wrong? And of the edges that it's getting wrong, being addressed by DCM?
So that's a really nice question. When you're missing 40% of the edges, you're basically missing just about everything. Not everything, but 40%, right? So it's not like you saying you're only missing the short edges. And one of the things that's happening with that particular simulation is that the trees are getting bigger, but all the edges have approximately the same length in that simulation. So it's not like you're getting all the edges that are big enough and only missing the ones that are small enough. But there is some theory that's been established for other distance-based methods that have the same theoretical convergence rate, where you can prove that every edge that's big enough, you will be able to reconstruct. But big enough is a function of how much error there is in your estimated distance matrix. So there are some theoretical results that can characterize the missing edges. For other methods, those results have not yet been able to be established for neighbor joining. But like I said, when you're missing 40% of the edges, it's not like you're only missing the short ones. Yes?
Yeah, you work, as far as I saw, in the Kimura two-parameter model and in the DTR model. In your slides, you mentioned both.
I also mentioned HKY.
Okay, but those are nucleotide-based models. Have you worked with codon-based models or other kind of evolutionary models?
Not in simulations. Actually, I take it back. We did actually in data that I did not show, we've actually done amino acid sequence evolution simulations as well. So we have simulation results as well in those models and theoretical results under amino acid models. So yeah, I mean, you can get the theoretical results under the other models, and you can get the simulation. We have done simulation results on amino acids as well. Comparable kinds of things show up. So these are not unique to DNA in terms of the patterns that we see.
Mm-hmm.
Eric?
You mentioned that reconstructing under simulated data is easier than real data, and I mean, that just indicates that there's a significant difference between the two, and I was just wondering how much you think reconstruction accuracy under simulated data indicates reconstruction accuracy for real data.
So to be clear, what I was saying was that it's not that the reconstruction accuracy was easier on simulated. I was saying that the running time and the way that the surface looked on the simulated data was less. I mean, so less running time was needed to get to a local optimum for simulated data. That's not a statement that we get an accurate tree. You know, it's just that the simulated data, you actually do get very close to an accurate tree, and it doesn't take very long to get there, and you don't get many, many local optima. Those are the kinds of things that you see. It does suggest that the model condition that generates real data is different from the model that we're generating the data. Now, what does that mean? For me, when I'm talking about theoretical guarantees, it's okay to stay inside the models, right? You're talking about theorems. You're talking about theoretical guarantees under Markov models. You can simulate under those Markov models, and you're happy. But if you want to make claims about what those results mean on real data, it does cause concern. You can say you can get the true tree from logarithmic number of sites, but that's under a certain model, and if that doesn't match reality, then that doesn't give you a lot of reason to be confident about reality. I think what it would be really interesting to do is to come up with what are the characteristics of data, of real data, that you want to make sure that your simulated data produce, so that you can start to have some confidence about your simulations being under appropriate models. And you know, that's something that I don't think people really yet know how to do. I mean, you'd like to match performance with respect to optimization surfaces, and you'd like to match other kinds of things as well, and I'm not sure what to do there, but it does make concern.
Okay, I think we'll stop
Thank
there,
you.
but thank you very much.